miRNA. A mature miRNA is 20 to 25 nucleotides long. It is formed from much longer precursor. It is endogenous RNA. It is developed from endogenous RNA molecule. The primary transcript is known as primary miRNA. Primary miRNA is processed in the nucleus and converted to 70 nucleotide long stem loop structure. The stem loop structure is called pre-mRNA. This pre-mRNA is transported to cytoplasm. The double-stranded portion of pre-mRNA is cleaved by dicer. This produces mature miRNA molecule. 1 to 6 miRNAs can be formed from single pre-miRNA precursor. Then miRNA is integrated into RISC complex, and from this point siRNA and miRNA share the same downstream pathway. The guide strand binds to complementary mRNA molecule. The bound mRNA is either degraded or silenced by some other processes. If the base complementarity is perfect, the mRNA is degraded by argonauts. Okay, so this is the same procedure afterwards, like siRNA. More commonly, base complementarity between miRNA and mRNA is imperfect. So even if the complementarity is imperfect, miRNA is partially bound to mRNA. So mRNA partially bound to miRNA cannot be translated. Double-stranded RNAs are never translated. Therefore, the genes are silenced. Now these are the differences between si and miRNA. siRNA pairs with target. So siRNA pairs with the target perfectly and induces mRNA cleavage of specific target. miRNA pairs with target imperfectly and can inhibit several targets having similar sequence. So miRNA are formed from transcription of sequences located either in between the genes or in introns. Human beings may synthesize more than 1,000 miRNAs. Nearly 40% are formed from introns, and these may target 60% of genes. Natural function of miRNA may be in regulation of gene expression. These miRNA may regulate tissue-specific expression and temporal expression patterns seen during development. Now I'll show you diagram. So this is the viruses. When they infect the cell, they inject their RNA into the cell. These RNAs are replicated and form secondary structures like the stem loop, and the stem loop, from the stem loop, pre-miRNAs are formed. These miRNAs are then cleaved by enzyme dicer or dosha, then exported out into the cytoplasm, cut with the dicer, and then they are introduced into the RISC complex. This is the pathway, RNAi pathway. So this is the inside the nucleus. The hairpin structure or stem loop structure RNA is formed. Dicer enzyme cut this double-stranded RNA into the short RNA fragments known as small interfering RNAs. These from the double-stranded siRNA, single strand of this siRNA is digested and the other strand is introduced into the RISC complex. This other strand is known as guide strand, which binds to the mRNA and then either modifies this mRNA sequence so that it is not translated, or cut the mRNA into small fragments so that the no proteins can be formed from it. Now these siRNAs can be derived from using PCR techniques, using forward and reverse primers. Synthetic siRNAs are then introduced into the cell with the help of a vector. So when the vector, along with the vector, this siRNA is introduced, in vitro transcription occurs. siRNAs are formed as like in vivo, and these siRNAs are introduced into the cells. I'll show you one more diagram. Okay, I'll post it on your group. Okay, so this was about RNA interference.